Yes.
Start.
Yes. Welcome to our zoology Zoom class for second session. Yesterday, we have completed myofibril, but not completed, I think. Some leftover is there, the remaining part. So we will start with the structure of myofibril again. Structure of myofibril. Myofibril is the one organelle, just like as cell organelle, myofibril also one organelle of the myofiber. Just you can compare normal cell. Normal cell contains nucleus and endoplasmic reticulum, mitochondria, and like this. In the same manner, the muscle fiber also one cell. It contains organelle, myofibril, sarcoplasm, and sarcoplasmic reticulum, mitochondria, and numerous nuclei. That's why we call it as syncytium, isn't it? Now, again, we start with the structure of myofibril or myofilament. This is, I think it is new for you. Yes. Each myofibril has alternate dark and light bands in it. Myofibril has two contractile proteins, actin and myosin. See, this is the myofibril. Already I told you, myofibril is the shape is cylinder and very long. Here, dark and light bands in it due to the proteins. Those proteins are actin and myosin. Here, actin is thin and myosin is thick. Are you able to see here? Here, there is no other color, only blue color is there. This blue color stripes are actin filaments. This zigzag line is Z line. This is actin filament and it is thin and light band. Otherwise, we can call it as I band. It is isotropic. So isotropic, already I told you, isotropic means it exhibits the properties, same properties in all directions. Yes, next one is, it is the dark one. The dark one is myosin filament. Here you can observe the red lines. Those are myosin and blue lines are actin. Here overlapping and little bit center of the pot is not overlapped. Only myosin is present. Due to the two kinds of proteins and thin nature of the myosin filament, it looks like dark in color. That's why we call it as a dark band. So alternatively they arranged. The dark band contains myosin and called as A band or anisotropic band. Isotropic, I explained. What is anisotropic? Anisotropic means it exhibits the properties different way in different directions. That is called anisotropic. Next, we will, already I discussed yesterday, this band is called I band. Because of the isotropic, we have given the name I band. Because of the anisotropic, we have given the A band. And the center of the I band, zigzag line is there, that is Z disc. And center of the A band, there is a one line is there, that is the M line. Yes, we will discuss again in detailed manner. See here. Yes. Thin filament, thick filament, actin and myosin. It is the I band, it is the A band, it is the I band again, and M line. M line is center of the A band, and Z band is Z disc is center of the I band. The I band is divided into two by a narrow line called Z line. It is an elastic fiber. Yes, we have to remember this word, elastic fiber. What is the nature of the elasticity? Elastic means it can stretchable, and after withdrawing the force, it comes to the normal position. So Z line only it has an elastic fiber, elastic nature. The thin filaments are attached to Z lines, so also called as Krauss membrane or Dobbe's line. See, the thin filaments, these are the thin filaments are attached to Z line. They are attached to the Z line. So this membrane is called Krauss membrane or Dobbe's line. The thick filaments are also held together in the middle of the A band by a thin fibrous membrane called M line. This is the M line. Center of the A band, one line is there, that is M line. It is a fibrous membrane. It doesn't have any elasticity nature. It is the fibrous membrane. Remember that when we are discussing the mechanism of the muscle contraction, it is important for us. M line is made up of myomycin protein. Myomycin. This is the myomycin. Myomycin protein. This is important for us. Next, here the portion of the myofibril between two successive Z lines is called sarcomere. One new word is there, sarcomere. See, this is one Z line, this blue color zigzag line. It is other Z line. In between these two portion of the myofibril, two successive Z lines, so side by side. In between these two Z lines, the portion of myofibril is called as sarcomere. Sarcomere is the structural functional unit of the muscle fiber or muscle tissue. Yes, muscle tissue. The portion of the myofibril between two successive Z lines is called sarcomere. It is the structural functional unit of the muscle tissue. This part here, this is the Z line here. See the arrow mark. Here, from here to here. This is this portion. This part of the myofibril is called sarcomere. A part of the A band without thin filaments is called H zone or Anson's disc. Yes, other zone is there. What is that? It is the H zone. Here, you are able to see here, it is the H zone. See here, H zone. Here you can recognize that there is no blue line. There is no, see here, there is no blue line here. Only the red lines, those red lines are myosin filaments. There is no actin filament. Only myosin filaments are present. Both sides, either side of the M line. This part is called here, see, this part is called, or we can write here, this part is called H zone, or we can call it as Hanson's disc. We will move to other Yes. Yes. Structure of contractile proteins. Already we discussed about them. Just I have mentioned the names of it. Structure of contractile proteins. Contractile means, again, they have the capacity to contract, come close. Yes, contractile proteins present in myofibril. There are two types. One is actin. Due to this structure and function of the actin and myosin, the muscle contraction is taken place. The actin filament is made up of 2F filamentous actin molecules. The actin filament is made up of 2F filamentous actin molecules helically wound around each other. See here, F actin is a polymer of a small protein called G-actin. See here, these are the spherical monomers. Monomer means a single one, single unit. This single unit is called monomer. This single unit is called monomer and it is globular. Globular means spherical, round shape, spherical shape. So each one called as monomer. The shape is spherical. That's why we call it as globular monomers or G-actin. These globular monomers polymerize and form the filament. What is polymerization? Polymerization means, for example, if you take only two monomers, they together form the dimer. In the same manner, if three means three of them together form the trimer. If several globular proteins or several globular monomers join together and form the more number of monomers join together and form the polymer. This phenomena is called polymerization. This phenomena is called polymerization. Due to polymerization, F-actin is formed. Filamentous actin is formed. So all globular monomers join together and form the filamentous actin. Next, we will go to, yes, two filamentous of tropomyosin protein also run close to the F-actin throughout their length. What is that? Tropomyosin. Two filamentous. Here also two filamentous. F filaments also two filaments helically wound around each other. Apart from these chains, other two chains also involved. Two filamentous of tropomyosin protein also run close to the F-actin throughout their length. Here, these are the F filaments, F filamentous actin. Other chain, two chains, these two chains. Apart from this, other two chains also run along with the F filaments. This is called tropomyosin. This is called tropomyosin. See, this is the tropomyosin. They also helically wound around each other. Next, other protein also is there. That is troponin. The troponin is a complex protein. It is not the just like chain. as tropomyosin or F filamentous. The troponin is complex protein. Complex means a group of is called complex. This troponin is distributed at regular intervals on the tropomyosin. This is the troponin complex. This is the troponin complex. Here also. Its structure is not chain-like. It is complex structure, and some complex structure means some subunits, maybe 2 or 3 or 4. Some subunits together form the complex. It is not the chain. Okay. So troponin complex, which molecules are, which subunits are involved in it? Let us move that concept. Tropomyosin and F-actin. This is the F-actin. This is the tropomyosin. See here, troponin. The troponin complex. Here, are you able to watch here? 3 ball-like structures. Here, 3 ball-like structures. So, troponin is made up of 3 polypeptide units. 3 polypeptide units. Those are TNT, TNI, TNC. What are those names? Those names has given according to their function. We can see here clearly. TNT attached to tropomyosin. These are the F-actin filaments, and these blue color and red color, these are tropomyosin chains. TNT. This is the TNT. See, this black color is attached to tropomyosin. Are you able to watch? Yes, I think you can. Here, this is TNT. Next one is TNI, this light blue color. What is the function of the TNI? Inhibits the myosin binding site on the actin. Inhibits the myosin binding site. Yes, each and every one has some special characters. In the same manner, here the F-actin has the active sites. Those active sites are sites which are the sites of binding sites of head of myosin. Those sites are masked by TNI. Inhibits. We can say that masked by TNI. The third polypeptide unit is TNC. TNC attached to calcium ions. So calcium ions are released in the sarcoplasm. In the sarcoplasm, from the sarcoplasm, the calcium ions move towards this TNC, this unit. Yes. So later we can discuss about it. This is the structure. Now I am explaining the structure only. Function we can say later, isn't it? So TNT, TNI, TNC, these 3 are polypeptide units. So the 3 polypeptide units joined together form the troponin protein. So troponin and tropomyosin are called regulatory proteins. Regulatory proteins. What does it mean? Regulatory proteins. Regulation means control. So whom they control? They control the mechanism of the muscle contraction. Yes, it has so much of important role. regulatory proteins, troponin and tropomyosin. Troponin and tropomyosin, they are able to mask the active sites of the actin filaments, and they are unmask the active sites of the actin filaments. Yes, you can understand later, but they are the regulatory proteins. They are able to mask and unmask the active sites. The most important other protein is there. That is, actin protein is connected to sarcolemma by dystrophin. Have you heard of this protein? Dystrophin protein? You might have heard of the disease name, dystrophy, muscle disease. The mass of the muscle is reduced and the number of symptoms it has. You might have read about it in paper also, in newspaper also. This is the dystrophin protein. Where does it located? Here, don't forget this. We are studying about the muscle fiber, that is muscle cell. Muscles inside the muscle cell, muscle fibers are arranged parallelly and sarcoplasm is there. In sarcoplasm only, muscle fibers are arranged, fibrils and are filaments, and then sarcoplasmic reticulum is there, and plasma membrane or sarcolemma is there. See here, just imagine, it is the muscle cell, it is the muscle fibril. Okay, this is sarcolemma. This dystrophin protein connected the actin filament with sarcolemma. It is the protein. This protein is controlled by actually gene. One gene, it is a specialized gene for the dystrophin. Yes, if the dystrophin is not function properly, we get the dystrophy disease. It has such a kind of importance, dystrophin protein. It is about the structure of the actin or arrangement of the actin. Next, we will go to other protein, that is myosin. It is also contractile protein. Myosin protein, otherwise called as motor protein. Why do we call it as motor protein? Because it is able to convert the chemical energy in the ATP into mechanical energy. That's why we call it as motor protein. Myosin. So actin is thick or thin. It is thin filament. Myosin filament is thick filament. Myosin filament is polymerized protein. It is also polymerized. Actin also polymerized and myosin also polymerized. Here, there we call it as globular monomers because of its shape. We have given that name, globular G-actin. Here, monomers, monomeric proteins called meromyosins. Their globular monomers, here meromyosins. It is the single unit. It is the meromyosin. Such a kind of meromyosins, several meromyosins together form myosin filament, thick filament. Okay. Two polypeptide chains wrapped around each other and constitute a myosin molecule. So it is meromyosin. It is a meromyosin. Meromyosin is form the myosin molecule, not the myosin protein. Don't get confusion. So number of myosin molecules together form the myosin protein. Each meromyosin, it is one meromyosin. Okay, are you able to understand? It is the one meromyosin. It is the structure of the one meromyosin. It is not the structure of the entire myosin protein. It is one meromyosin. Such kind of meromyosins, number of meromyosins involved to form the myosin protein. Two polypeptide chains wrapped around each other and constitute a myosin molecule. Each meromyosin has two important parts. We are able to see here two important parts. It is globular head, globular again, so round, globular head with the short arm, it is the short arm, and tail. Two important parts it has. Globular head composed of heavy meromyosin, HMM, heavy meromyosin. The tail is made up of light meromyosin. The head has other two sides. One is actin binding site, another one is ATP binding sites. Their importance you are able to understand during the explanation of the muscle contraction. Actin binding site, actually this site bind or attached to actin. It is the actin binding site. ATP binding site. Already, as I told you, motor protein, it converts the chemical energy, the ATP, into mechanical energy. So ATP added to this site, and ATP is converted into as ADP and phosphate, inorganic phosphate, and release the energy. Later we will discuss all those things. But here now I am explaining the structure. So two sides. They have the two sides. One side is actin binding site, another side is ATP binding site. Yes, I want to clear the location of the meromyosin. That's why I have given again this image to you. It is the sarcomere. You know it. It is a structural functional unit of the muscle tissue. Z line, actin, proteins. They are connected with the Z lines. The center of the sarcomere, it is the myosin protein. Yes. In the previous image, we have seen the meromyosin structure, head and tails. How they arranged. See here, the specific arrangement is there. That's why I am explaining here. This is the head. It is the tail. This is the head. It is the tail. So, have you identified here? All the heads are facing towards Z line, one side. Even this side also, all the heads are facing towards this Z line. So, like this, the heads are oriented towards the Z lines. The tails facing towards M line. This is the M line. Yes. This is a specific arrangement. Why does it need such kind of arrangement? The heads has the binding site of actin. That's why they have to bind the actin. So actin filament is here. There is no actin filament. That's why all the heads facing this side and this side. In the center of the myosin is concentrated with the tails. Yes, I hope you understood the actual location of the meromyosins in myosin protein. I want to give one more image to you, because muscle fibril or filament is cylindrical, isn't it? So here see, it is also cylindrical shape. These are the heads around it, heads. So actual structure is like this. It is the sarcomere. I think you might have understood, isn't it? Yes. So far, we crossed a number of names of the proteins. What are those? Actin, myosin, tropomyosin, troponin. But I want to give two more names, titin and nebulin. These two also one kind of proteins. So altogether, myofibril contain 6 types of protein. 6 types of proteins. Actin and myosin. These are the contractile proteins. Already we discussed it. Contractile means they are able to contract. And tropomyosin and troponin, they are the regulatory proteins. They are able to regulate the mechanism of the muscle contraction. And accessory proteins. Accessory proteins are titin and nebulin. Where do they locate? Where do they arrange? What are the functions of titin and nebulin? Have you heard of those names, titin and nebulin? If you do, so far if you don't, just you remember. See, I want to show the 6 types of proteins arrangement. It is the sarcomere. Always we take one sarcomere because it is the structural functional unit of the muscle tissue. Actin is connected with Z line. This is actin connected with Z line. This is actin connected with Z line. This is actin connected with the Z line. The center of the sarcomere, myosin. Does it connected with Z line or not? In generally when we are talking about, we never talk about the myosins are connected with Z lines. But see here connection. There is a connection. That connection is, moreover, that connection is linked this side Z line to other Z line through the myosin protein. This protein is called titin. This protein is called titin. What is the function of it? Titin provides elasticity and stabilizes myosin. Yes, titin protein gives the elasticity and stabilizes the myosin. Okay. Next, actin. Already we have discussed about the structure and components of the actin. It has the tropomyosin and troponin proteins also. But other protein, see here, this is a blue color. This is nebulin. Nebulin helps in align actin. Align means in line, straight line. So it is the nebulin. The nebulin is connected to Z line and actin, and it pass on throughout the actin protein. Here also you remember that titin also not only connecting of the Z line and myosin, it pass on through the myosin till the other Z line. So that's why they are able to maintain the stabilization, stable, firm structure. They are able to move. They are able to move, and they are able to produce the contraction and relaxation, even though they maintain their structure with stability due to the presence of titin and nebulin proteins. These are the functions of the nebulin and protein. Already actin and myosin and troponin and tropomyosin already we have discussed. These are the 6 types of proteins in myofiber. Yes, just I want to do the comparison. Actin and myosin, because these two are the regulatory proteins. That's why, not regulatory, sorry, contractile proteins, actin and myosin are. I want to compare them. present in the light bands. I think you know, you can compare. You can compare on your notebook without watching this also, because already you have understood the structure of the actin and myosin completely. You can write present in the light bands. It is present in the dark band. Actin is isotropic band, I band. Myosin is anisotropic band, A band. These are the thin filaments. These are the thick filaments. An elastic fiber, Z line, is present at the center of the each I band, which holds the thin filaments. Here, it is not the elastic, it is fibrous. Fibrous membrane, M line, is present at the center of each A band, which holds the thick filaments. These are the points to compare. If you get more points, you can include the points, because always you people are dynamic and vibrant, because you are the students. Next, another concept. So far we have completed the structure of contractile proteins and then other proteins also. And now we are going to triad system. The concept triad system. What is triad system? Are you able to find out what is this one? This is the muscle cell. Section of the muscle cell. It is plasma membrane or sarcolemma. Actually, when we are talking about the cell, just we draw this like this, isn't it? It is the cell membrane and nucleus and then endoplasmic reticulum or mitochondria. Organelles, all organelles we write like this. In the same manner, here also it is the sarcolemma, and inside, that means in the sarcoplasm, it is sarcoplasmic reticulum. This is the sarcoplasmic reticulum, and these are the fibrils, muscle fibrils, and somewhere here you cannot see here, somewhere mitochondria and number of nuclei very near to the sarcolemma. It is the structure of the muscle cell. Here, one special character is there. What is that special character? Sarcolemma invaginates. This is the invagination. Invagination means go through into deep. Invaginates into the sarcoplasm and forms transverse tubules. Transverse tubules. This is the not longitudinal. It is a transverse tubule. Yes. It has started from where? From the sarcolemma. Sarcolemma, just it is the sarcolemma. It invages like this. This is invagination. And form the T tubule. Why do we call it as T tubule? Because it is a transverse tubule. So T tubule. This is the T tubule. Next, what is this one? Sarcoplasmic reticulum. This sarcoplasmic reticulum, ends of the sarcoplasmic reticulum, there is a cistern. Cistern means cistern. What is cistern? Have you heard of this word? Cistern? Cistern means one cavity or space in between two membranes. That space is act as a reservoir. That is called cistern. For example, Golgi complex. Golgi complex also have the cistern. Two membranes, in between there is a space. So here you can observe here completely. It is sarcoplasmic reticulum. Ends of the sarcoplasmic reticulum, it forms cistern. It is the just like it act as a reservoir. There is a space in between two membranes. Okay. Now, when we call it as triad, the T tubule and the two terminal cisternae at its sides form the triad system. Yes. T tubule, it is the T tubule, it is a cisternae and it is also cisternae. Either side of T tubule, cisternae are present. These three of them, these three structures, three of them together form triad system. I think it is a 2 marks question in your examinations. It is the triad system. Are you able to follow? I hope you can, isn't it? See here. It is the sarcoplasmic reticulum. These are, what about this? These are the actin and myosin filaments. You remember entire the cell when we are talking about one single organelle, but you can imagine the remaining organelles also present in this sarcoplasm. See, these are the actins and myosin filaments. Next, we go to other concept, that is, I want to say one more thing here. Most of the stored calcium of the sarcocyte is located in the terminal cisternae. Yes, this cisternae, already I told you that cisternae, the function of the cisternae is it acts as the reservoir, something stored in it. What will store? Calcium ions. The calcium ions stored in this cisternae, calcium of sarcocyte. Cyte means cell, sarcocyte, that means muscle cell. So calcium is stored in it. Already I told you calcium is important mineral for muscle contraction and proper functioning of the muscle. So we need more calcium. Next, other concept, that is motor unit. What is motor unit? A motor neuron and the set of muscle fibers innervated means supply by all the telodendrites constitute a motor unit. See, this is the neuron, motor neuron. This motor neuron, these are the endpoints, telodendrites. These telodendrites reach to sarcolemma or muscle fiber. This is the muscle fiber. This is the sarcolemma. This is called motor unit. Junction between a motor neuron and the sarcolemma of a muscle fiber is called the neuromuscular junction or motor end plate. Here you can see that this is the telodendrites and lemma, sarcolemma. Here we can see the just directly they are contacted, linked, but it is not linked. Here you can see that there is a gap. It is the knob of the telodendrite. It is the sarcolemma of the muscle fiber. There is a gap. This gap is called synaptic cleft. The entire this area is called neuromuscular junction, and it is also called as motor end plate. Again, we can talk about in this concept during the explanation of the neuromuscular activity. Next, assignment. Describe the structure of skeletal muscle. Write the differences between actin and myosin. Already we have written. Draw the neat label diagram of the ultrastructure of muscle fiber. You know it. Draw the diagram of sarcomere of a skeletal muscle showing different regions. Yes, those are very familiar to you. What is triad system? Very simple. Okay, thank you.